Chapter 6, Section 2, the process of meiosis. During meiosis, those diploid cells are going to undergo two cell divisions that result in haploid cells. Cells will go through 2 rounds of division in meiosis. This is going to reduce the chromosome number and create genetic diversity, which is so important for the sexual genetic diversity that we need. So meiosis will produce genetically unique cells. It's going to result in haploid cells, and it takes place at only certain times in an organism's life cycle. Meiosis 1 and meiosis 2 each have 4 phases, very similar to those that are found in mitosis. Pairs of homologous chromosomes will separate in meiosis 1. Homologous chromosomes are similar, but not identical. Sister chromatids divide in meiosis 2. Those sister chromatids are copies of the same chromosome. So remember, during interphase, we made a copy of all of our DNA. And so what happened was, each of those individual chromosomes produced an exact copy of itself. And those stuck together, and we call those sister chromatids. Now, those homologous chromosomes are, remember, like the right sock, left sock. They're similar, but not identical. So there you can see the diagram. Meiosis 1 occurs after DNA has been replicated. Meiosis 1 divides those homologous chromosomes in 4 phases. During prophase 1, the nuclear membrane will break down, the centromeres and the centrioles begin to move, spindle fibers attach and assemble, the duplicated chromosomes will condense, and the homologous chromosomes begin to pair up, so we're in that organization phase. During metaphase 1, spindle fibers will align the homologous chromosomes along the cell equator. Each side of the equator has chromosomes from both parents. Anaphase 1, the paired chromosomes will separate from each other and move towards opposite sides of the cell. Sister chromatids remain attached. We call that little structure where you see the little 4 ends, that's called a tetrad. During telophase 1, the spindle fibers disassemble and the cell undergoes cytokinesis. So all of this sounds very similar to mitosis. But one of the things that occurs somewhere in here is, because there's so much DNA crammed into such a small area, sometimes we have this process called crossing over. And so that's how you sometimes get a mix of genetics from your mother and your father. Occasionally, these chromosomes are so packed tightly, they will actually swap sections of genes with one another. So you get to be a combination of your parents' genetics. This is why siblings oftentimes will look similar, but they'll have little differences. I went to high school with four boys. There were 4 boys in the family. Mom had blonde hair and blue eyes. The dad had brown hair and brown eyes. The oldest boy had blonde hair and blue eyes. The next oldest boy had brown hair and brown eyes. And then the next boy had brown hair, blue eyes, and then finally the blonde hair and blue eyes. Or sorry, the blonde hair and brown eyes. So every possible combination they could have got from their parents, those boys exhibited. Meiosis 2 divides sister chromatids in 4 phases. So we're not going to stop and take time doing interphase again. We're going to jump directly into meiosis 2. So the DNA is not replicated again. We're going to pick up right where we left off. In prophase 2, the centrosomes and centrioles move to opposite sides of the cell. Spindle fibers begin to assemble. Metaphase 2, those spindle fibers align along the chromosomes, along the cell equator, and they pull them apart. In the end, we end up at telophase 2 with nuclear membranes forming again around chromosomes. The spindle fibers break apart, and we have 4 mismatched cells. They are not identical to the parent. It's a combination of genetics that could go either way. They could favor mom, they could favor dad, they could be a completely 100% split. This is going to differ from mitosis in several significant ways. The first is that meiosis has 2 cell divisions, while mitosis only has 1. In mitosis, the homologous chromosomes never pair up. They never have a need to. Meiosis results in haploid cells. Mitosis results in diploid cells. Haploid cells are going to develop into mature gametes. So these are the cells, the sperm that are ready to fertilize an egg, or an egg that's ready to be fertilized. We call that process gametogenesis, and that is simply the production of gametes. Gametogenesis differs between males and females. Sperm in spermatogenesis will become streamlined and motile. The sperm primarily contribute DNA to an embryo. There's not a whole lot else there. So that's all they're really contributing to that fertilized egg that they're getting ready to fertilize. Eggs, however, during oogenesis, it's a funny word, but immature egg cells are called oocytes, and they go through oogenesis, and they're forming eggs that are going to contribute not only the DNA, but they're also responsible for providing the cytoplasm and the organelles to the embryo. During meiosis, the egg gets most of the contents, and the other cells are going to form these things we call polar bodies. They just get reabsorbed back into the system. They don't really serve a purpose. It's just the byproduct of what we're trying to produce here. So that egg is very important because it's very large. It has to house everything that growing embryo will need after fertilization.